Now we're going to go over Chapter 6, which is photosynthesis and how we acquire energy from the sun. So all of the energy used by almost all living cells is ultimately coming from the sun. We have plants, algae, and some species of bacteria that are able to capture sunlight and the energy that is contained in it through a process called photosynthesis. But of all of those rays that come to us from the sun, only about 1% of the available energy in sunlight will actually be captured through photosynthesis. So when we're thinking about that, we're thinking of a photosynthesizing organism like this tree. We're zooming into the leaf, into a specific cell within that organism, and then all the way into this organelle, the chloroplast. This is going to be necessary for any kind of photosynthesis that's going to take place, whether it's algae or plants. We're going to be inside this chloroplast, and everything's going to take place either in the thylakoids or the stroma of that chloroplast. So the chloroplast contains these internal membranes called thylakoids. So this individual piece is a thylakoid. When we stack all of these thylakoids together on top of each other and talk about that column, that's actually called a grana. So we're talking about the grana if we're talking about a whole column of thylakoids, but really all of those processes are going to happen with, all of those processes for photosynthesis are going to happen either in the thylakoid or in this semi-fluid area that the thylakoids float in, which is known as the stroma. Our photosynthesis can be summarized in these three basic stages. So the first stage is just capturing the energy from sunlight. So we could think of that as the moment that the light interacts with the photosynthesizing organism, the moment the light hits a leaf. Next, we're using the captured energy and water to produce ATP, which is energy, and NADPH. We have not talked about NADPH yet. But what you need to know is just basically that the main purpose is going to be to either donate an electron or a proton. That's the main reason that this is going to show up. So we're going to need that. We're going to need both energy and we're going to need a donated electron in order to finally make the sugar at the end of our photosynthesis process. But this big piece of photosynthesis is happening in the thylakoids. So that's happening in the membrane of the thylakoid. And these are light-dependent processes. We also have our third stage. In the third stage, we're using that ATP and NADPH that we made in our light-dependent processes in the thylakoid. And we're combining them with CO2 from the atmosphere. And that's what's allowing the plant or the algae to make those carbohydrates. These parts are known as light-independent reactions because they do not need light, they do not rely on light, and they happen in the stroma of a chloroplast. So what does NADPH stand for? It stands for nicotinamide adenine dinucleotide phosphate hydrogen. But you don't need to remember that. We're not really focusing on remembering really long names for no particular reason. Any biologist I've talked to has referred to this as NADPH, and that's how we're going to talk about it for the rest of the course as well. But I didn't want to continue without at least telling you what those letters are standing for. So our NADPH is useful for donating electrons and hydrogen, protons, to the reactions that are catalyzed by enzymes. In this purpose, we are donating electrons for the purpose of photosynthesis. So our chloroplasts are present in plants and they use NADPH as a part of the pathway that's making those sugars from the sunlight and carbon dioxide. When we're looking at the reaction for photosynthesis, this is the summary that's given in our book. So we can see we've got 6 molecules of CO2, carbon dioxide, 12 molecules of water combining with the energy from light to create our products, glucose, water, and 6 molecules of oxygen. I included this reaction because I wanted to show you guys one that kind of brought these images in to make everything stick a little more, because you really do need to know what are the main reactants going into photosynthesis and what are the main products that are being made in photosynthesis. But you might notice a difference when you compare it to this reaction from the book. And that's just because this water that we're showing on the product side isn't necessarily being made by this reaction. It's left over, so it's kind of waste. You can see that 12 molecules of water went into the reaction and 6 molecules are left over at the end. So we didn't make water here. It's just the excess water that was not needed for our reaction. What we did make is this glucose, which is the main goal of photosynthesis. Plants are trying to make glucose. They need it for their cellulose. They need it for the starch in their roots. They need it for all of the fats and lipids and proteins that are going to be produced. So this is the main goal of photosynthesis. And oxygen is the super useful byproduct that we benefit from that plants have to get rid of. So what are the two main reactants? Carbon dioxide and water. We're going to need both of those in order to be able to complete photosynthesis. And then light energy as well. Our main products are glucose and oxygen. So our photosynthesis is divided into two types of reactions. We touched on this briefly, but we're going to go more in depth now. The first is the light-dependent reactions. These reactions take place only in the presence of light, and that is going to happen in the thylakoids of our chloroplasts. The result of this is the production of ATP, energy, and NADPH, our electron donor. Next, we have our light-independent reactions. So think independent. It does not need light. These reactions are going to take place in the stroma of the chloroplast, and they will result in that final formation of our organic molecule, our glucose, our sugars. This is more commonly referred to as the Calvin cycle. So that's what's happening in the stroma when we're making our sugars. That's the Calvin cycle. Here you can see that happening. So here we've got our light reactions in our thylakoid. It's really mostly taking place in the membrane. The light is going to interact with that thylakoid membrane. We are also going to take up water into the thylakoid membrane. And as a result, that water is going to be split so that we can actually get the hydrogen that we are needed for this NADPH molecule. But as a result, we don't end up with the right balance of hydrogen and oxygen left over to just have water molecules. So we're going to have an excess of oxygen molecules because we've used our hydrogen. It's part of this product. But now we have a surplus of oxygen. So that's why these plants are getting rid of oxygen, because it's left over from the product they're trying to make, NADPH. That NADPH is needed, along with ATP, to feed into the Calvin cycle in the stroma and produce the sugars. So our NADPH and our ATP are going to combine with the CO2 from the atmosphere in the stroma. This is the light-independent part of our reactions because no light is needed for this part to take place. And that's what's going to produce our sugars at the end of photosynthesis. When we're talking about this light from the sun, it's really comprised of packets of energy, and we call those packets of energy and light photons. So our sunlight has photons of varying energy levels, and you can see those reflected here. We've got radioactive elements with a really high frequency, intense, high energy, and then we've also got light that is able to be perceived by the human eye. So here's our range of visible light. And that's in this kind of wavelength range. And then as the wavelength peaks get further and further apart from each other, our photon energy is decreasing. So you can think of this as like higher energy, excited. Think of it almost as vibrating. It's very high energy whenever it's got these really close peaks. And then as the wavelengths get further and further apart, they're slowing down. This is like more like microwave frequencies, radio wavelengths for FM and AM radio. So they're much slower and much lower energy on this side of the spectrum. This is what we use. It's called the electromagnetic spectrum. This is what we're using to represent the range of visible light. So of all of this range of photons, only between 400 nanometers and 740 nanometers are we able to actually perceive that light and actually see the photons that are showing up or being absorbed or reflected. Whenever we're talking about pigments, we're really just talking about molecules that are able to absorb light energy. Oh, let's go back. So our pigments, normally that comes up in conversations with art. You've heard about really high pigment paints that are super expensive and high quality. But a pigment is just anything that's able to absorb light energy. And therefore, it's also able to express some kind of light energy that we are able to perceive. But the pigment in human eyes is only absorbing photons of intermediate energy levels. So that's this range, the range of visible light. That's what we're able to pick up on and perceive. There are photosystems that are embedded in the thylakoid membrane, and they contain these pigments. So the primary pigment molecule in most photosystems is chlorophyll, and this pigment acts as an antenna to capture energy from the sunlight. So when we think of the purpose of an antenna, like when we're trying to get better cell reception, or when we're trying to get better reception on our radio, when we're trying to listen to the radio, and so we have an antenna, we're funneling those waves to a specific point. That's the goal of the antenna, to give a point where those wavelengths are more likely to interact. And so that's the same thing with these pigments. These chlorophyll bundle together to a point that is going to be more concentrated, where the photons will be funneled to. Also, when that photon interacts with the chlorophyll, those individual chlorophyll pigments are going to pass the captured energy and an excited electron from the photon between each other. So kind of imagine it like hot potato. They're passing this highly energized electron from the photon, from the energy of the sun, back and forth to each other until it can finally be donated for the next part of the photosynthesis process. So our main pigment is chlorophyll, and chlorophyll absorbs light at the end of the visible spectrum. So that's like the reds and the blues, and that's why it reflects green, because it's absorbing blue and red light, but it's not absorbing the green, it's reflecting it. And that's why it shows up as green whenever we look at plants. So we see chlorophyll as green. And there are two versions of chlorophyll that are present in plants. There's chlorophyll A and chlorophyll B. We should know that we have those two forms of chlorophyll. We don't need to know much more detail about what separates them at this part of our process in learning. But there are also accessory pigments that absorb light of different wavelengths that are not captured by chlorophyll. So here's another example of one of those pigments, carotenoids. And in the last slide, you could see it was represented by this yellow leaf here. That's because carotenoids will absorb green light, but they will reflect red, orange, and yellow light. Let's look at it this way. So here we have a graph showing the wavelengths relative to the light absorption for these different pigments. You can see chlorophyll A here. It absorbs violet and blue light, and that's shown by the peaks here. So this looks kind of red to us on the map, but or on the graph, but I'm imagining it's supposed to be slightly further towards this violet end of the spectrum, because the peaks are showing us where those pigments are absorbing. That means they're reflecting what's in this range, what's not in the peak area. Also, our carotenoids are peaking in, here's the carotenoids. So they're peaking in blue and green, which means our carotenoids are absorbing blue and green, because that's where that peak is happening for absorption. And that also means that they're reflecting everything else over here. So our carotenoids are reflecting yellow, orange, and red light, which makes sense why the leaf color changes in the fall, because in the fall we have depleted levels of chlorophyll because as a result of the colder temperatures. And as we have less and less chlorophyll, our plants are reflecting less and less green. But the carotenoids are pigments that are less common in plants and also less affected by the drop in temperature. So as our chlorophyll reduces and it's not reflecting as much green, suddenly the carotenoids are representing the color for the plant because they are reflecting back red, orange, and yellow, and they're no longer covered up by the really strong chlorophyll reflection of green. This is just to touch on photons and pigments so we can get a basic idea of how they're contributing to the process of photosynthesis. But don't get too hung up on this. We do not need to remember the specific ranges for carotenoids in chlorophyll A and chlorophyll B. You don't need to know which ones absorb which and remember that for the rest of your life. This is just a basic overview because we are using this information as how it interacts with photosynthesis. So these are the pigments that are interacting in photosynthesis. In plants, our light-dependent reactions are going to take place in those thylakoid membranes that we talked about inside the chloroplast, here. So here's one thylakoid, and now here we're zoomed in on the membrane of that thylakoid. You can see the stroma on the outside, the thylakoid space, so the inside of the thylakoid is shown on this side. Our pigments and protein molecules are also occurring as complexes called photosystems. So these photosystems are working together to process the photons that are coming in from our sunlight, to process that energy. This is one look at a photosystem. So we're in the thylakoid membrane, and what we're calling the photosystem is really just the system of chlorophyll that's passing along this energized electron from our photon. So let's look at that a little more. Our photon is going to interact with chlorophyll, and it's going to excite or energize one electron. That excited electron from the photon is going to be passed around, kind of like hot potato, like we said before, between these different chlorophyll pigments. Only once it reaches this specific chlorophyll pigment, which is called the reaction center chlorophyll, will it be able to actually pass that excited, energized electron on to an electron acceptor. And when we say electron acceptor, we literally just mean anything that is able to accept an electron. So whatever is there and available to make a bond with that electron. Because we got rid of that electron in this system, we also need an electron donor. And that's where water comes in. When we break up those water molecules, we have an excess electron that can be donated here. And then we're able to get rid of the excess oxygen left over because we can expel oxygen through the cell membrane that will then be expelled through the stomata of plant leaves. But the goal is pass the energized electron on through until it can get to the electron transport system, which is the next stage of photosynthesis. Once that electron's passed through, we have to replace it with an electron donor that's coming from water. So our excited electron is shuttled along, and this is, don't let this change in imagery fool you. This is the same process. So here we're still in the thylakoid membrane. This is still the area where we have photosystems 1 and 2. You can see photosystem 1 and 2. And then our electron transport chains are here. But the excited electron is going to be shuttled along a series of electron carrier molecules that are embedded in the protein. So here's our carrier molecules that the electron is passing through. The energy from that pumps hydrogen ions, which are protons, across the membrane. So that's creating this flow of hydrogen ions across the membrane. The hydrogen ions are going to diffuse back across the membrane through ATP synthase channels here. So we're bringing a high concentration of hydrogen ions in, And that's what's going to allow for the consistent flow of hydrogen ions back out through this ATP synthase channel. At that point, we are capturing energy to combine ADP with an inorganic phosphate, which is how we make ATP, which just means energy has been produced. And if you'll remember, our second stage of photosynthesis, the goal is to produce ATP and NADPH. So after ATP has been made, the electron is going to leave the electron transport system and enter another photosystem where it will be re-energized by the absorption of another photon of light. Our electron is shuttled through a second electron transport system at that time. No ATP is made, but that's when we get NADPH. It will be formed from NAD+. NADPH is going to be used as the electron donor in the Kelvin cycle. So that means we've made ATP and we've made NADPH, and both of those are what we need to combine with carbon dioxide in the Kelvin cycle. But let's look at that here. So we want to see it all kind of in a flow, so from beginning to end. To start with, don't get too hung up on the specific, all the different names for all of the different molecules that are going into this process. What I want you guys to understand is the flow of the process and what's being produced at each point. So the first interaction for photosynthesis is going to happen at photosystem 2. After that, we go to the electron transport system. After that, we go to photosystem 1. And after that, we have another run through an electron transport system that's going to result in NADPH. This can be confusing. The fact that photosystem 2 is the first stop in photosynthesis does not seem like very intuitive. You would expect to start at photosystem 1. But that's really just because scientists first discovered photosystem 1. So the first thing they noticed was this interaction, and they named it photosystem 1, expecting to stumble upon other photosystems later on. They did, so some years went by, they discovered photosystem 2, but they still at that time didn't really know which came first, which makes sense because this is a cycle, and when you're looking at a cycle, it's hard to determine a beginning. That's like determining the beginning of a circle. So they kept them in this place until a couple years after that, whenever they realized, okay, photosystem 2 is the logical way to talk about the beginning of photosynthesis, because that's where the first photon interaction happens, and that's where the water splits so that that electron can be provided. So that's whenever they realized photosystem 2 really is the beginning of this process, but people have been talking about photosystem 1 and photosystem 2 for years at this point, so they didn't want to rename them and confuse everybody even more. So they just left the names as they already were, but kept them in the order that they actually need to be thought of. So that's why photosystem 2 is our first stop, just because it was discovered a little late. But when we're looking at photosystem 2, that's where we are capturing a photon of light and releasing that excited electron through the reaction center chlorophyll. So here's our excited electron that is being passed on to the electron transport system. It's also where our water is being split to provide that donor electron where one was just passed through. And then that's where our byproduct of oxygen comes into place as well. So there is where our oxygen results from the splitting of that water molecule, which will then be released out of the cell and eventually out of the surface of the leaf through the stomata. That's photosystem 2. Then our energized electron is passing through the electron transport system, and that flow is also creating a proton gradient to form a higher concentration of hydrogen ions inside the thylakoid space. As a result of that high concentration of hydrogen ions in the thylakoid space, we have hydrogens that are being pumped out of our ATP synthase channel. That's what's happening here. And when that happens, they provide the right conditions for ADP to phosphorylate into ATP here. So that's where our energy is made in our first electron transport system. The energy is released into the stoma so it can be used in the Kelvin cycle. Our electron continues. So now we've got an electron that has landed in our photosystem 1 along with a new photon, and that is going to go on to become an energized electron that will be passed on to another electron transport system. But the goal of this second electron transport system is not to make ATP, it's to use those hydrogen ions and attach them to NAD+, so we have NADPH, which will be an electron donor in the Kelvin cycle. So that's kind of an overview of how we are making ATP and NADPH in the thylakoids, and you'll need to keep in mind that these are the light-dependent reactions. So this is the part where we need light for these reactions to take place. So we're starting at photosystem 2. Its reaction center consists of more than 10 transmembrane proteins. So even though we're simplifying it, I need you guys to keep in mind that there are a lot of transmembrane proteins going in to make these reactions possible for everything to come and go across this membrane. It is surrounded by an antenna complex of pigments, and they are funneling those captured photons to the reaction center, because the reaction center chlorophyll is the only one that can pass on those energized electrons and donate them to the electron transport system, which is where they need to go. The reaction center yields an excited electron to the primary electron acceptor. So the primary electron acceptor is going to take that excited electron, and also water will be split to provide that replacement electron to the reaction center once it's donated the excited electron from the photon. And that's what's going to result in the production of oxygen. So that's how plants are making oxygen. It's just they're throwing away what they didn't use when they broke up a water molecule to get the electrons. So that's photosystem 2. That's what's just happening here. The photon is interacting, exciting an electron. The excited electron eventually makes it to the reaction center chlorophyll. Water splitting enzymes will split water molecules so that we have a byproduct of oxygen, and we are able to donate that electron to replace the one that is passed onto the electron transport system. The electron transport system will receive the excited electron from the electron acceptor, and the electron transport system is comprised of proteins that are embedded in that thylakoid membrane. So all of this is happening consecutively in that thylakoid membrane. One of these proteins is going to act as a proton pump, and that's moving a proton from the stroma into the thylakoid space. Because we know hydrogen ions are coming into the thylakoid space, and a bunch of them are going out because of the concentration gradient through ATP synthase. At the end of the electron transport system, the electron is passed to the reaction center of photosystem 1. Remember, photosystem 1 is the second in order for this process. So now we come here to photosystem 1. We've got our ATP production that just took place as a result of those hydrogen ions coming through our ATP synthase channel. We've got a new photon interacting with chlorophyll and exciting another electron. That electron is going to be donated to the electron transport system. And that's what's going to provide the chance for NADP plus to join with a hydrogen ion to form NADPH, which is needed in the Kelvin cycle. But when we're talking about this, I do want to take a moment to mention chemiosmosis, because that's the name for what's happening during part of this process. So chemiosmosis is really what's happening here. Anytime we are phosphorylating ADP to ATP and creating that energy, that is part of chemiosmosis. In chemiosmosis, the thylakoid membrane here is impermeable to protons. So protons can't just pass through freely. And protons can only re-enter the stroma by traveling through a protein channel called that ATP synthase channel. As the protons diffuse back down their concentration gradient and pass through the ATP synthase channel, which is facilitated diffusion, ADP is phosphorylated into ATP, and that is chemiosmosis. I tried to think of a simpler way to summarize that, and this is the simplest sentence I could come up with. It's the movement, so chemiosmosis is the movement of ions across a membrane to produce energy for cellular processes. So it's just kind of creating that flow. It's creating that movement of ions, and as a result, ADP is able to phosphorylate into ATP. And then we've made energy for our cellular processes. So now we're looking at the same process. We still have photosystem 2, our electron transport system, that ATP synthase channel that's happening at the end of our beginning electron transport system. So we're looking at the same process. We're just really going over this repetitively to make sure that we can remember all of the different steps through photosynthesis. So as 4 electrons are removed from 2 water molecules, here's our water molecules, we have removed 4 electrons, oxygen will be released. The excited electron in the electron transport system reaches the proton pump protein embedded in the thylakoid membrane. That's here. So this is the proton pump protein. And the energy of the electron is used by this protein to pump a proton into the thylakoid space from the stroma. So that's how we're getting all of these hydrogen ions in, through that protein. And it's a pump, it's active because we're going against the gradient. So we're bringing all of these hydrogen ions in until we have a really high concentration of hydrogen ions. And then at that point, they're going to naturally want to diffuse because they're following their concentration gradient. And they'll use this facilitated diffusion channel called ATP synthase. So these protons that are pumped into the thylakoid space are going to be taken out into the stroma through that ATP synthase channel. And the movement of this channel is what's actually providing the chance for ADP to become ATP. Bam. Another protein in the membrane will carry the energy depleted electron. So here, these lows are kind of indicating how it's becoming the lower energy electron as it's traveling along. Carries the energy depleted electron to photosystem 1. The top surface of photosystem 1 here is embedded with that antenna complex containing the pigment molecules. So these are all of the chlorophyll pigment molecules, and we're calling that the antenna because that's what's pulling these photons to it. A photon will strike the chlorophyll molecule, and the energy will be passed on until the electron is excited again, excited like energized, and ready to leave photosystem 1. Once that excited electron leaves photosystem 1, that is what's going to trigger the electron transport chain and produce NADPH. So now we have this provided electron, which has made it possible for our hydrogen ion to combine with NAD+, and now we have NADPH. So in photosystem 1, our first stop, our second stop, we are receiving the electron from the first electron transport system, and we're also absorbing another photon of light. The reaction center is going to yield the excited electron to another electron transport system, and that's going to use the electrons and the hydrogen ions to form NADPH. So because this removes a proton from the stroma with those hydrogen ions, the production of NADPH is also helping to establish that proton gradient so chemiosmosis can continue. And chemiosmosis, remember, is just turning ADP into ATP. So you can see all of that happening here. We come to photosystem 2, excite the electron. It will be passed between these chlorophyll pigments until it reaches the reaction center chlorophyll. At that time, a water splitting enzyme will split our water molecule so we can replace the electron that's about to be sent away. Oxygen is the byproduct that will be released at that time. Our excited electron is being taken to the electron transport system, which is going to help create our proton gradient. That proton gradient inside the thylakoid space is going to then follow facilitated diffusion out of the ATP synthase channel, which is going to allow for ADP to become ATP, so we've made energy. At that point, our electron comes in, and another photon from light is going to also interact with photosystem 1 until an electron can be an excited electron, meaning high energy, with energy from the sun, can be carried onto the next electron transport system we're going to interact with. At that time, our NAD+ can combine with our hydrogen ions as a result of this excited electron, and we have formed NADPH. So at that point, we have both of the molecules that we need to supply the Calvin cycle with what it needs once it combines them with carbon dioxide. So our synthesis of organic molecules, and again, organic molecules is just molecules containing carbon. So the synthesis of organic molecules from CO2 employs the Calvin cycle. That's what we're talking about with the cycle that's happening in the stroma. These reactions are also known as C3 photosynthesis. I've never heard a scientist talk about them that way, but this is in every book, so I included it as well. So this is another term for the Calvin cycle. I'm still going to refer to it as the Calvin cycle. But that Calvin cycle has to turn or complete a rotation 6 times in order to form one glucose molecule. And that's because we're getting 1 carbon from every completion of the cycle. So once we get to 6 completions, then we have 6 carbons for our glucose molecule. The products of our light-dependent reactions, everything we just talked about, photosystem 2 to our electron transport system, to photosystem 1 to our second electron transport system, all of that is our light-dependent reactions. And they produced ATP and NADPH. Our ATP is the energy that drives the Calvin cycle, and our NADPH provides those hydrogen atoms, which are going to be the necessary electrons that are needed to create our glucose molecule. So we have this breakdown here, but I actually found a video that kind of shows it in a more step-by-step way, so I think that this is better. But I do want to point out, the video is going to refer to these names as well, and I don't want you guys to get hung up on trying to panic about remembering those names. It's irrelevant to us. You don't need to remember those names. We're trying to understand photosynthesis. So please just go with the flow of the video. Don't try and remember those names. We don't need to know them. But I do want you guys to see the kind of breakdown of how the Calvin cycle is producing these sugars. Reactions of photosynthesis. Steps. The light reactions and the Calvin cycle reactions. In this tutorial, the photosynthesis and Calvin cycle reactions. They allow us to So that's our overview of the Calvin cycle and the processes that are combining ATP, NADPH, and carbon dioxide from the atmosphere to create our glucose. Interestingly, there's also a mechanism that plants have to pause photosynthesis. And this is something that they do, especially when it's hot outside and they're having a hard time creating those sugars. And it's kind of like, imagine a plant that's just trying to survive the day and the heat is not going to be focused on making sugars to store energy for another time. Instead, they're just trying to survive. So at that point in time, plants will close the openings in their leaves, which are called stomata. If you're just talking about one of them, they call it stoma. But they would close those openings in order to prevent water loss. The closed stoma is also preventing gas exchange. So that's how water gets in and out of the leaves. That's also how CO2 goes into leaves and how oxygen leaves them. So if we've closed our stomata, we don't have any gas exchange, no CO2 is coming in, no oxygen is leaving. As a result, oxygen levels are going to build up inside the leaves, and we're going to get less and less concentration of CO2, which plants need. As a result, that enzyme, rubisco, which normally binds to CO2, is going to start binding to oxygen instead, because there's a super high concentration of oxygen because none of it has been able to leave. And this process is called photorespiration. So it's kind of like a pause on photosynthesis. We're not able to produce any more sugars right now. We're trying to survive this heat. We're closing the stomata so no gas exchange can happen. And here's an example of what that looks like. So these are the stomata, if we're talking about them plural. Once they close, oxygen's not able to leave. Water's not able to get out of the plant either. CO2 is not able to go in if these stomata are closed. And as a result, it's allowing our plant to thrive in a high oxygen containing environment. So it's basically shifting the energy. So now the plant's not trying to make sugars, it's trying to break down whatever toxic compounds are built up within it. So it's like a pause so that the plant can switch gears. And instead, this metabolic pathway, photorespiration, is going to recycle phosphoglycolate, which is a toxic compound to plants that they naturally acquire. And that's going to transform phosphoglycolate back to phosphoglycerate. When that happens, oxygen substitutes for carbon dioxide in that first reaction of our photosynthetic carbon fixation, the Calvin cycle. So basically, now we're using our built-up oxygen to get rid of toxins that are in the plant instead of using a bunch of energy to continue making sugars when it's super hot outside and the plant can't sustain that action. So this is a really interesting way that they're able to pause photosynthetic processes and kind of switch and change up and divert to getting rid of toxins within the leaves. That is our general overview of photosynthesis, but I'd really like to go through it in a consecutive linear way like this. So I've kind of put this summary together. This slide is going to have all of our light-dependent reactions and our main summary of what they're producing. The next slide will have our light-independent reactions that are happening in the stroma. So first, our light-dependent reactions. Light-dependent reactions happen in the thylakoid, mostly in the membrane. These reactions are using water and light to produce ATP, which is the energy that we need, and NADPH, which will function as our electron donor because it provides that hydrogen that can donate an electron. These are needed to combine with carbon dioxide later in the Calvin cycle to make sugars. So when we're talking about these light-dependent reactions, it starts when an energized photon is passed through photosystem 2, travels through the electron transport system, which results in the production of ATP. Next, the electron transport will continue through photosystem 1 and into another electron transport system, which results in the production of NADPH. So at that point, ATP and NADPH have been made. They will leave the thylakoid and enter the stroma of the chloroplast. So now we're coming out of the thylakoid into the stroma, where we are available for the process of the Calvin cycle. The Calvin cycle is part of our light-independent reactions. It's happening in the stroma. It does not need light to take place. These reactions are going to take up that ATP and NADPH that were made in the thylakoids and use them alongside CO2 from the atmosphere to create the sugars. And that's our entire photosynthesis process. So that's our summary. I want you guys to be able to remember what happens in the light-dependent reactions, what happens in the light-independent reactions, what are the products of each of those reactions, and of photosynthesis as a whole. So you should know what goes into photosynthesis and what's produced by photosynthesis. If you have any questions, feel free to reach out.